Hello student, how are you? Myself Pawan Kumar Mishra. Today I am again with you to explain the next topic related to the sexual reproduction in the human being. In the previous two lectures, you completed male reproductive system and female reproductive system. Now, the purpose of male and female reproductive system, what is the main function of their primary organs? You know very well. The main purpose of their primary organs to produce male or female gametes, right? So there is the gametogenesis. So gametogenesis is a process. Gametogenesis is a process of formation of male or female gametes with the help of gonads by meiosis. So you can say generally gametogenesis one type of process of formation of gametes or sex cells by meiosis. This is the major division here. By meiosis, okay, in germ cell, of gonads. This is the simple definition of the gametogenesis, you know. Right now, there is the one word is the gonad. So you know for the gametogenesis there are the two gonad. The first one you can say testis, and second gonad ovary. You know testis in male, you know this is the primary organ of the male. Ovary in female, this is both are primary organ, right? So first of all, formation of sperm, male gamete, there is the sperm or spermatozoa. This sperm, another name, spermatozoa, right? Formation takes place in the testis, and then this process is known as spermatogenesis. So gametogenesis, the first type of gametogenesis is spermatogenesis. If gametogenesis takes place in male, then this is known as spermatogenesis. But when it takes place in female, from the ovary they form ovum, right? Then this process is known as oogenesis. Right now, the process gametogenesis again divided two types: spermatogenesis and second one oogenesis. In oogenesis, there is the formation of ovum. This is the gamete or sex cells in female, and in male sperm, this is the gamete or sex cells in male. So first of all, we have to study here one by one, right? What is spermatogenesis and what is oogenesis? First of all, I am going to explain the spermatogenesis here. The first most important topic is spermatogenesis. You know, spermatogenesis means formation of sperm or spermatozoa, right, from testis in male by meiosis in spermatogonia, right? So in simply you can say spermatogenesis is a process in which sperm formation takes place inside the testis. Right now, This spermatogenesis, you can say, take 64 days, about 63 or 64 days, right? It will take 64 days about. In spermatogenesis, the main organ, it occurs in, you can say, it occurs in testis. Testis is the singular form. Tests are the singular, plural form for it. It occurs in the testis. You know very well what is the testis. Inside the testis, there is seminiferous tubules. Seminiferous tubules. These are the structural and functional unit of the testis. Right now, these seminiferous tubules, site for, this is the site for spermatogenesis. This is site for spermatogenesis. This is the very most important question. Write which one acts like the site for spermatogenesis? Seminiferous tubules. If you observe, in seminiferous tubules, inner lining of seminiferous tubules right, have two type of cell. Two type of cells. The first cell, you can say germ cell or primary germ cell. Okay, right? And second one, supporting cells. These supporting cells is also known as Sertoli cells. Right? So there is the two type. If you observe, I will also explain with the help of diagram. First you know the flow chart. Right? So first, inner lining of the seminiferous tubules. In a tube-like structure, there is a cavity inside. So from the cavity, the first layer, that is the inner lining of seminiferous tubules. Right? Suppose that this is the tube-like structure, this one. Right? So this one represent inner lining, this one inner lining. Right now, so from inside, this represent the inner lining of the tubule. So inner lining of seminiferous tubules have two type of cells. First, primary germ cell or germ cell. Second, supporting cells. These germ cells, there is the meiosis, mitotic division first. Mitotic division. When there is the mitotic division, so these primary germ cells form large number of spermatogonia. They form large number of spermatogonia. What is the spermatogonia? Spermatogonia is known as sperm mother cell. Another name you can say sperm mother cell. Sperm mother cell. Right? These spermatogonia, if you observe the next flow chart, right? These spermatogonia, after that, these spermatogonia, this one. So first from primary germ cell they form large number of spermatogonia. So this is, you can divide this phase as multiplication phase. Multiplication phase. You can also divide gametogenesis in various phases, in which the first phase is multiplication phase. right? In which primary germ cell by the mitotic division they form large number of spermatogonia or sperm mother cell. After that, these sperm mother cell or you can say spermatogonia. You know these germ cells have diploid in the nature 2n. Spermatogonia also diploid 2n. Right? These sperm mother cells, few of them, grow in size. Few cells of the spermatogonia grow in their size, becomes large, and then they are known as primary spermatocyte. When few cells of spermatogonia grow in size, right, then they known as primary spermatocyte. They are also diploid in nature 2n. So from here to here, right, this is the growth phase. This represents the growth phase. If you observe, this one is the growth phase. So first phase is the multiplication phase in the gametogenesis or spermatogenesis. Second phase is the growth phase, right, in which spermatogonia grow in size and ultimately few cells change in the form of primary spermatocytes. These primary spermatocytes undergo meiosis first. They are the first meiosis division. You know very well, in the first meiosis, number of chromosomes become just half. So from one, there is one, suppose that there is the one, from one primary spermatocyte cells, if there is the meiosis first, so there is the formation of two, right? From first, you can say, there is the formation of two secondary spermatocyte cells. From one primary spermatocytes, there is a formation of two secondary spermatocyte cells. In the secondary, there is a number of chromosome is n. You can say if there is the 44 autosomes plus, suppose that XY sex chromosome present in this cell. So there is a two cells formation, and in one cell there are 22 autosomes plus any one sex chromosome, X chromosome. Other cell 22 autosomes plus other sex chromosome, Y sex chromosome. So from one primary spermatocyte, there is the formation of two secondary spermatocytes by the meiosis. After this, so this is haploid number of chromosome becomes half in secondary spermatocyte. Then there is the meiosis second. Meiosis second is like the simple mitotic division. So each cell form two cells, but number of chromosomes remains same here. Number of chromosomes always remains same here, right? In this you can form four spermatid. There is the formation of four spermatid, right? Four spermatid. There is the four cells. 1, 2, 3, 4. It 22 autosomes plus X, 22 autosomes plus X, 22 autosomes plus Y, 22 autosomes plus Y. So you can observe there is the formation of four spermatid here, right? Then what happened? Formation of four spermatid now. So from primary spermatocyte to spermatid, this one. This is maturation phase. You can say this is the maturation phase. Right now, this one is the maturation phase. This represents maturation phase from this one, primary spermatocyte to fourth spermatid. This is the maturation phase. Maturation phase. So first phase you know very well, when germ cell change in the spermatogonia, this is the multiplication phase. The second phase, when spermatogonia change in spermatocyte, this is the growth phase. And when primary spermatocyte ultimately change in fourth spermatid, then this represents maturation phase. After the maturation phase, there is, you can say, differentiation phase. These four spermatid, this one, these four spermatid, right? I am putting here. These four spermatid, after that, they change in the form of four sperm, or you can say spermatozoa, right? They change in the form of four sperm, or in other words you can say spermatozoa. So what happened? This process is known as differentiation. This process is known as differentiation. What happened here? What happened in the differentiation here? First we have to know, right? What happened in the differentiation? First of all, suppose that these such type of structure right now, right? First they form the sperm. If you see the simple diagram of sperm, so you get such type of structure. Right now, this part, blue color part, this represent acrosome, right? And this black color, this part represent nucleus, right? After that, if you observe, there is a small leg, and then you can observe the middle piece. I will explain the diagram of this later, don't worry. Right now, and there is a spiral strip of mitochondria. So first here, the first in the differentiation, what happened? Formation of acrosome. In differentiation, first one, formation of acrosome. Formation of acrosome, the first point here. The second, this nucleus enlarge. Okay. Third point, you can say formation of axial filament. This one is known as axial filament. Axial filament. And this one is the centriole. Both points known as centriole. So for separation of centriole, right? Fourth point, this red color is spiral mitochondria, So there is the formation of spiral mitochondria, right? Both centriole get separated here. Acrosome formation, elongated proper nucleus, this is upper is the proximal centriole, and then lower is the distal centriole. From distal centriole there is the formation of XII filament, formation of spiral mitochondrial in the middle piece. This one is the middle piece. This one represents the middle piece. For these, this is the differentiation. This cell ultimately change in this form. This is known as differentiation. Then they are completely known as sperm. And for this spermatid should attach. It means spermatid get attached to Sertoli cells. Sertoli cells, right? Pyramidal shaped Sertoli cells provide nourishment to the spermatid, by which spermatid ultimately, by following changes, they ultimately differentiate in the form of this sperm. So there is the 4 cells, 1, 2, 3, 4. Each cell, each spermatid get differentiated in sperm. So there is the formation of 4 sperm. And this process is known as, this differentiation, this process is known as the process in which spermatid change in the form of sperm. This process is known as spermiogenesis. This is the given in your NCERT. Spermiogenesis. What happened? In spermiogenesis, spermatid get differentiated into sperms. Right? After that, if you observe these sperms which are formed here, these sperms, there is another process, spermiation. By spermiation, sperms get released from seminiferous tubules, and Sertoli cells. Because you know, spermatids get attached to the Sertoli cells. These are the pyramidal cells, Sertoli cells, right? Spermatid get attached here, and when spermatid change in the sperm after maturation, these sperm comes out from the seminiferous tubules and also detached from the Sertoli cells, and this whole process is known as spermiation. So in this complete process, the total time taken is 63 or 64 days for the spermatogenesis right now, and the process in which there is the development of sperm from spermatogonia or germ cells which present in seminiferous tubules is known as spermatogenesis. How can we represent this spermatogenesis with the help of diagram, which is very, very most important from the CBSE board examination? You listen here. It's a very simple. First of all, there is this line represent epithelial or general epithelial of seminiferous tubules. Right? On the inner side, there are many cells present. There are some pyramidal cells present like this, and you can draw a large pyramidal cells here, in this way. This is the large pyramidal cells represent Sertoli cells, right? Okay, and there is primary germ cell. There is the primary germ cell, this one. there are many cells here, right? This is the germ cells. Germ cells. Few germ cells, you can say they form large number of germ cells here, right? You can say this one is the spermatogonia. Spermatogonia. These spermatogonia, few spermatogonia enlarge. This one, they enlarge. When they enlarge, then they are known as primary spermatocyte. You know, the germ cell is 2n, spermatogonia is also 2n, right? This primary spermatocyte is 2n diploid. In this primary spermatocyte, when there is the meiosis first, so they form, after meiosis first, they form secondary spermatocyte cells. Secondary spermatocyte cells, right? This one, the secondary spermatocyte cells. Which have haploid number of chromosomes, just half, n. These secondary spermatocyte cells, after meiosis second, they form spermatids. And these spermatids get attached so spermatid is also haploid. Spermatid get attached to Sertoli cells. This is the Sertoli cells are nursing cells, you can say. Sertoli cells are nursing cells which provide nourishment to the developing embryo. This is known as Sertoli cells right now. So these Sertoli cells provide nourishment, and after that they ultimately change in the form of sperms. They ultimately change in the form of sperms, you can say. Ultimately they change in the form of sperms. Right now, so these finally form this one, sperm, right? So this also you can simplify a diagram. You can draw the diagram in on the basis of their ploidy also, how spermatogenesis takes place in the seminiferous tubules. This is the seminiferous tubules, you can say. Okay, seminiferous tubules. This is written here. So this is the complete spermatogenesis. I hope all the points you understand very well. Thank you very much.